Welcome, Takema, to the Collaborative Research Center Origin and Function of Meta-Organisms here in Kiel. It's a great pleasure to have you here. You are one of the leading figures in the symbiosis field. You give a talk in the international conference. Following up that in a few minutes, you are a researcher at the National Academy Institute in Japan. You're a professor at Tsukuba University, but you are most well known as the research director of a similar collaborative research center worldwide known as the Fukatsu Evolving Symbiosis Project and funded by the Japanese government. You have more than 350 papers, and it seems that all what you are looking for is insects and the symbionts in the insects. And how did that all evolve in yourself when you think back in your childhood, or how did you find the insect so fascinating? And then somehow that must have evolved into your fascinating and very original research question. Can you tell us a bit how that happened?
Okay. First, Thomas, thank you for inviting me to this very exciting opportunity. And this is my virtually first time to visit Germany. So, yeah, I'm so excited. And for your question, yes, I'm biologist. Especially, I define myself as evolutionary biologist. Yeah, that means that I would like to understand, not understand. Initially, I was fascinated by biodiversity. There are so many insect creatures. When I was a small child, really I loved insects so much.
Who told you that? Your parents? The school?
No, that was maybe imprinted in my DNA.
Imprinted in the DNA?
Yeah, maybe.
Yeah, probably. yeah.
I'm not sure, but anyway, when I was 2 or 3 years, I really chased many insects. There's a grassland there, and I crawling into the grassland and find a small insect. But actually, I was born and bred in Tokyo. Tokyo is a big city and no forest. Yeah, that is very environmentally boring place. But anyway, even in Tokyo, there are many small creatures. When I find some vegetation, and that was I was fascinated. And then, so when I get older and older, yeah, some kids are fascinated by insect transiently, and they forget it. But I never forget it. I grow up and go to the university. At that time, yeah, really, I already decided that I would be biologist. Yeah. But yes, I was. Yeah, I studied many things, like molecular biology, developmental biology, but I was really fascinated by the evolution. That explains why so huge diversity of organisms are created, and also the origin must be the single event because I myself, insects and plants, everything is have DNA, cell, and they are living by producing the enzyme and catalyzes all the biological reactions in the cell, and that is the same. So that is because so when we pick up the human our own DNA, put it into the bacteria and produce something. That is the biotechnology. But that is possible because all the organisms have the common ancestry. That is amazing. Yeah, and especially when I was initially fascinated by insects, insect diversity. But when I go to the graduate course, and my professor was Dr. Ishikawa. He was a pioneer of insect symbiosis in Japan. And in his professor's room, I found a book. That is a book of Paul Buchner. Yes, he is a very famous German microbiologist who is a true pioneer of symbiosis research. His age was the early 20s. At the time, no molecular biology, no biotechnology. The cutting edge biological technology was microscopy of card size. Yeah, that is the reason why in Germany at the time, microbiology developed at a very high level. And at the time, many German and some French and European researchers observed everything in the world using the microscope. And some of them found that not all, but many insects actually are not simply an insect, but inside the body and the cell, there are microorganisms, and they are living as a symbiotic entity. Yeah, that is that's why in his book, very incredibly diverse such symbiotic associations and described, and I was surprised. I thought the insects are insects because they are insects. Yeah, butterflies, beetles. It's right. Well, I'm quite familiar with them since my childhood. But actually, they are not the simple insects, but they contain other entity and living together. And I was fascinated by the symbiosis, and that is why I started to work on symbiosis so extensively.
I see. Yeah, thank you so much. It's, now we see the picture much clearer, and your fascination about insects. The word you said about Paul Buchner, who certainly was one of the major, probably the founder of the whole field, and limited technology, that is certainly also true, but he made fantastic observations which are valid today. I also have his book in my office. If I see back in your early papers, it follows that up by careful observation of at a microscopic level of these interactions between this insect, which is a meta-organism because it has many other organisms associated with. But time has changed now, and you are also leading this change with new technologies. And now we have most sophisticated sequencing technologies, culturing technologies, all these omics, etc. How important was that to get more insight, or is it just a technological something which is nice to have? How important was this technological progress?
Technology is important, but technology is a technology. The technology is important, and the aspect is everybody can use it once it develops, and so easy. And so, the researcher who firstly invented the technology is highly appreciated. Some of them get a Nobel Prize. But the followers are just using technology, like we use the iPhone. Yep, that is very easy, very convenient, but it's open for everybody. Here, it is difficult to find the originality of the work. That is just a tool. So the way the originality decide is an important point. Yeah.
I fully agree, and this is, it fits very well to your how I see you. The point is, yes, it's only technology, and the question behind that is the important point. Nevertheless, I would not tune down the importance of technology too much, because look to your own papers. I mean, they got so much deeper now because you could do all this genome analysis, etc. You have your specific question, which you want to answer. But I think at the level where we are right now, and also the symbiosis field, and probably also that's why many young people are getting attracted to the field, because we are using fancy technology, fancy iPhones, and that many people find attractive. But you have to give them then also a very original question. So I would say, yes, technology was a major part in progress of the field. Would you agree?
Yes.
Yeah. Okay. Yeah. The other feature when I look in your work is that you just go out, maybe here in the hotel park, you find a beetle, you look at it, or another insect, and then maybe you find it interesting, and all of a sudden you have a new model for something. That is unusual. So we have these so-called model organisms, which is Drosophila and the mouse and the elegans. You are you're playing around with all kind of different organisms, which are maybe in some extent more difficult to study because they are not yet worked out genetics and blah, blah, blah. How important are models as defined, Mouse C. elegans, Drosophila, maybe some fish, versus non-models? Or is that comparison actually makes not too much sense because any animal can be a model?
Yeah, sure. So that is a very important question, but in my opinion, yes, to do some new things. Of course, for using mouse, Drosophila melanogaster C. elegans, there has a very fancy techniques are available for them, but everybody that is so too competitive. Everybody are working on it, so I don't like such a situation. I would like to do something by my own approach and my own pace. And also I would like to work on what is truly interesting for me. Then, so the important point is, yes, there are so many biodiversity. Fascinating phenomena are omnipresent. Everywhere we can find it. But it is not easy to work on it experimentally, biologically, and so important point is what I first try is when I find some interesting insect, try to keep it in my laboratory. And in this, and in many cases it doesn't work, but sometimes we maybe successfully keep it in the laboratory. Then it will be a new model only for me. That is my way of thinking and how I proceed with such a system and why I'm working on so diverse insect-microbe symbiotic systems.
Thank you so much. In 2019, you wrote a paper in Frontiers in Microbiology with the title Grand Challenges to Launching an Ideal Platform for Publishing Microbe Insect Symbiosis Studies. And my question is, this was 2019.
Yes, but at the time, that is not, because at the time I was appointed by a chief editor of the microbial symbiosis section of the journal. And so, as the chief editor, I would like to invite the submission of the insect-microbe symbiosis field researchers. And so, I wrote the editorial. So that is just such an invitation message, and that's not so philosophical implication.
So then I ask you today, what are the challenges in your field?
In my field? That is okay. Maybe today I will talk about it. Yes. What is the challenge? Evolution. I say that I'm evolutionary biologist, but some people say evolutionary biology is not actually real science because it's something occurred in the past, and we or you are speculating what happened in the past, but you do not see the evolutionary directly. That's such an inference, a speculation, deduction. But then I thought that I would like to directly observe the evolution in front of my eyes, on my laboratory bench.
In real time.
In real time. Yeah. And I started to establish the experimental evolution of symbiosis, such a system in the laboratory. And recently, we successfully done it. And that is the basis of what you mentioned, my ERATO project, Evolving Symbiosis project. Now, in my laboratory, Escherichia coli, very famous model bacterium, is evolving into insect mutualists, one after another. Yeah,
very fascinating. We're very much looking forward to what you have to say as new on that. It again uses all your progress uses state-of-the-art technology. manipulating microbes, genetically engineering microbes, etc.
Yes.
So again, I see, the original question, and this is again an original question, can we do now evolution in real time? But it has to go hand in hand with an available technology that you can study deep. Would you agree?
Yes, of course. And also, the big challenge is, yes, in case of many symbiotic systems, the symbiotic microorganisms are uncultivable because they are highly specialized for the life within the host, and many metabolites are supplied, and the environment is highly constant because of the host homeostasis, and so. Uncultivable means that they cannot be genetically not engineered. So, working on symbiosis, symbiotic microorganisms has been very difficult, challenging. But then also, we are now trying to clone the whole genome of the microbe in the baker's yeast, and then manipulate their genome is now genetically manipulatable, not directly, but within the yeast cell. And we established such system to circumvent the conventional difficulty of microbial fastidiousness.
Yeah, fascinating. You're a very successful researcher. You're a model case. I mean, you have a large team. You have
model case.
You are. You are, and you have a wonderful team, young people, enthusiastic. I met them. You collaborate with many different colleagues in Japan from different areas. You are able to raise their interest to work with you on insects and stink bugs, and also they are coming from different fields. That's really an achievement. If you would, I ask you now, what would you tell the young generation which is watching you, which is maybe admiring you when they see you giving talks or being on TV or seeing your movies? Everything goes so smooth and it's so easy, and it and that's so I want to become a scientist. I want to become a biologist. I'm sure it was not easy, and you told us it is not easy. But what would you tell an early career scientist? What does it need to make a successful career in academia? Not necessarily only in symbiosis, but what does it need when you look back to your own success?
That is not easy to answer because there is no general answer. Yes, science, research, and life is specific to each personality, and also so. But anyway, in my case, I'm very crazily fascinated to insect and biodiversity, and in many cases, such people going to the field of science, for example, taxonomy or ecology or such the macroscopic research field. And also some scientists go to the molecular biology and genomics. But in my case, but the special, I'm quite exceptional in that I have a very strong background of macroscopic and biodiversity aspect of biology, but at the same time, I'm very interested in the molecular biology and genomics and developmental biology. So, how to say, I'm quite multidisciplinary. In many cases, such multidisciplinarity is realized by the collaboration of the multiple researchers. But if it is integrated into one single personality, it is much more strong. So what I suggest to young generation is do, learn, practice everything for yourself. Don't make limits.
Yeah, very
that
good.
Is my message.
Yeah, very good. Touching also an important field means being able to talk to different people, multidisciplinarity or interdisciplinarity. I think this is certainly, certainly very important.
It is easy to speak out, but it is not easy to practice.
Exactly.
Yeah.
Yeah. And symbiosis field, per definition, has experts from very different disciplines, and so, thank you so much. We're coming to an end. I would like to ask you, if you look a bit ahead of time, where do you think your research will be in 5 years or in 10 years, or just what will be understood in 5 years or 10 years, and what are still open major questions? Where will the field go in the next few years, in your opinion?
There are many aspects. Of course, the more ambitious purpose is creating the living entity from the nothing. De novo creation of the life is one of the probably most challenging and feasible, practically feasible. Maybe it may be difficult, but anyway, on the earth, the life emerged from nothing. Then really, in my case, I successfully established the symbiosis, mutualism between E. coli and stink bug insect in the laboratory. But before I did it, nobody saw that such a thing is possible. But it was possible. Then I expected in near future, probably somebody will create the life, something looks like life form from the non-life. Then it will be a breakthrough of the next big stream of the next biology. That is what I'm imagining. That is, I'm not sure it is 5 years ahead or 10 years ahead and 100 years ahead. But anyway, that is I'm sure that will open a new big stream of biology. Then we know only one form of life, DNA, RNA, and ribosome, and such this system. But there must be other systems, actually, but we don't know. We have never seen it, and I would like to see it. But maybe when I'm alive, I cannot see it. But anyway, in future, somebody will do it, I believe.
Okay, wonderful to close with a lot of bold ideas. I think you started with bold ideas, and the future of science and the future of progress depends on bold ideas and to get deep answers. Thank you very much for coming.
Yeah, and thank you for providing me such an exciting and very nice opportunity.
Thank you.